Hello, everybody. Welcome back. We are going to do a little bit of Chapter 8 material, so take a look in the syllabus and look at the specific page numbers to cover. We're not going to do the whole chapter. So this first little mini lesson is just an overview, kind of going over what recombinant DNA technology is and kind of an illustrative example of the three main goals of recombinant DNA technology. So first of all, recombinant DNA technology is just recombining DNA. So it is using technology to modify the genes that are found on certain chromosomes. So they could be chromosomes in human cells, plant cells, other animal cells, or in bacterial chromosomes. So we are recombining the genetic information of typically two different organisms, and they don't even have to be of the same domain. So we could take human genes and bacterial genes or plant and animal genes, or there's lots of different combinations. That's what researchers are working on, is seeing, can we get some beneficial outcomes now that we have some better understanding of how DNA works and how we can manipulate it to make improvements, hopefully, for humankind. So in this example, the three goals of recombinant DNA technology are down here in the bottom boxes right here. So eliminate undesirable phenotypic traits, create beneficial phenotypic combinations, and produce vaccines, antibiotics, hormones, or enzymes. So removing bad things, producing new beneficial traits, or producing a product, something that can be used by people, pretty much. So we are going to track how this could potentially be done in an example using, let's just say, insulin. So the gene to make insulin. So diabetics prior to genetic recombination technology used cow and pig insulin, I think mainly pig insulin for diabetics to be able to regulate their blood glucose levels, which works, but it's not very good because it's not human insulin. So once this kind of technology was more commonplace and could be done pretty well and pretty safely, they were able to actually allow or get bacteria to produce human insulin that could then be extracted from these big tanks of bacteria and processed and isolated, and so we don't get any bacteria bits in there. It's just the pure human insulin, and then that is what diabetics are using now. So let's take a look at that. So let's say on our this would be our gene of interest. So this is human chromosome, right? So that purple is a human chromosome. And then let's say our gene of interest is insulin. Okay, so what researchers can do, and we're going to talk about the tools that they use to do this a little bit later on in the other video, is they can actually go in and cut out that gene of interest. They use tools like restriction enzymes to come in and just chop on either side. And so this green part of the gene, if that was expressed, it would make insulin. Okay, so that's our goal. What they can then do is coax a plasmid to take up that piece of DNA. We saw bacteria do it all the time in horizontal gene transfer. They're just recreating that same process happening in the lab. So we insert the human insulin gene into a plasmid, and then we allow the bacteria to take up the plasmid. That's this over here. That's just transformation. So environmental DNA that's picked up by a bacteria and is able to express those particular genes. So now we have a bacteria that we can grow in test tubes that has the ability to produce human insulin. And so we grow it, and that would be kind of going down this path. Harvest proteins coded for by the genes, which would be insulin is the hormone. Okay. So that's one way that we could do recombinant DNA technology, is producing a product that could be used by humans. Other examples of this, eliminating undesirable phenotypic traits. They're working on that with sickle cell and some other blood disorders, is gene therapy, going in and inserting a healthy form of the hemoglobin gene into patients' bone marrow. So they are now making proper hemoglobin instead of the mutated misfolded hemoglobin. So the people who would have been dealing with signs and symptoms of sickle cell are no longer feeling those effects because they have healthy hemoglobin. And creating beneficial combinations of traits that could be, there's an example I usually talk about of golden rice. So a lot of times if you see like food donations to developing countries, they have big bags of rice. And so the citizens get the rice and it gives them food. And so if they're in a famine, they are allowed to have some food. But the problem with rice is it's really good, it's starchy, it's got a lot of carbohydrates, but they're not getting a complete, nutritional food. There's not a lack of vitamins. And one of the main vitamins that people who are living in really poor conditions, they lack vitamin A. And vitamin A is really good for eyesight. It helps to make your retinol so your rods and cones work properly. So one of the side effects of severe malnutrition is blindness. And so some researchers were like, okay, so we can ship them this rice, but is there a way that we can incorporate vitamin A in rice? Because rice typically doesn't produce vitamin A. It's produced in orange vegetables like carrots and squash and things like that. So what they were able to do is to figure out the metabolic pathway in these orange vegetables, isolate the genes that code for that, splice them into the rice, and when the rice grows, the rice grains themselves, instead of being white, they're kind of an orange. So they're called golden rice. So when individuals eat the rice, not only are they getting the calories from the starchy component of the rice grain, but they're also getting vitamin A. So you might hear this of genetically modified foods. That's kind of been in the headlines lately. That genetically modified foods is a byproduct of recombinant DNA technology. All right, so that's the overview, the three goals, what researchers are trying to accomplish with this, these techniques, these tools and techniques. So then the image that I will end on for this little mini lesson is maybe someday we can recombine the genes of some interesting animals. We've got a owl with a cougar, a chameleon with a cow, and a rhino with a chipmunk. I think the rhino chipmunk is my favorite, but they're all pretty cool. So just as something a little funny. All right, I will see you next time, and we will talk about tools and techniques that researchers use to accomplish some of those goals. All right, I'll see you next time.